Welcome, everybody. It is a pleasure today to start the 17th webinar of the online edition of the Protein Electrostatic Conference. The speaker of today is António Batista from the Institute of Chemical and Biological Technology, António Xavier in Portugal. So António graduated in biochemistry from University of Lisbon in 1988. Then he got a PhD degree in biochemistry in 1998. In 2014, he became principal investigator at the Instituto de Tecnologia Química and Biologica. Among his interests, there are electrostatic processes in protein, especially constant pH molecular dynamics. And he will be talking today about how to address the effect of pH on protein dimerization, a constant pH molecular dynamic study of beta-lactoglobulin. After his talk, we will discuss a little bit on a topic which raised some interest in the present webinars, which is at which point are we in the computationally estimating the charge of the biomolecule. So António, it's a pleasure to have you here. You can start your presentation.
Mm-hmm. Okay, thank you, Walter. So let me share the screen. Can you see my presentation?
Yes.
Okay, great. So hello everyone. Good morning, good afternoon, good evening, whatever it is for you. I'm going today I'm going to talk about a study of dimerization of this small protein beta-lactoglobulin and use it as sorry, just give me a second because sorry, I'm trying to move my Zoom controls out of the way, but yeah, anyway. I will present the study of this dimerization of this protein using constant pH MD. I mean, in the past we applied constant pH MD to many different types of processes, from protein misfolding to peptide membrane interactions, to membrane proteins as well, and I mean to a lot of different things really. But until now we haven't addressed the processes with protein association. So we decided to turn our attention onto that. So I mean, for this audience, this slide is not really needed, I think. But anyway, we have all good reason to believe that protein-protein association changes protonation states, and we have evidence of that from for instance, in terms of pKa shifts. We know that pKa shifts can be shifted. I mean, at least at the experimental level, there are some studies that study pK shifts due to the binding of small ligands at the computational level. We also have much evidence of that, mostly using more simplified methods than the one I'm going to show today. But in any case, so we decided to study our constant. Basically, what I'm going to show you is to try to, okay, we can use constant pH MD to study anything, but if we are studying protein-protein association, what is the best way to look at the results and to try to integrate things with each other? So it's more or less the aim is more or less that one. So as a kind of test system, we use this small protein, beta-lactoglobulin. It's a small protein, 162 amino acids, which is the most abundant in milk whey. Not in humans because we don't have it, but in most mammals, particularly in cattle. I mean, this protein has been studied since the 1950s at least, and it was, I mean, studied with a lot of different biophysical methods in particular. So this was a kind of benchmark system in those days. So there's a lot of data for this system. As I said, it's a small protein. It's mostly a beta viral with an additional strand and one helix. And interestingly, the function of this protein remains unknown. It's not clear what it does. It has a hydrophobic pocket that can bind small molecules, hydrophobic molecules, but we don't really know what is its function. It can be a carrier of these molecules. In some people suggest that, okay, it's present in milk just for effects of nutrition, because it has a rather balanced content of amino acids. So it's not really clear what its biological role is in cattle, for example. But anyway, for us, it has several practical relevance, like, I mean, in terms of the milk processing in industry, and it also can cause allergies in humans, and there have been some attempts to try to engineer it to work as a drug carrier. So this protein has some interesting pH-dependent features. One of them is the so-called TAN4 transition, which has been observed originally by Charles Stanford a long time ago, and it's believed to take place around 7.5 pH. It's a quite smooth transition, actually, but I mean, the midpoint is more or less around 7.5. It's believed to be related nowadays with the motion of a loop, the so-called EF loop, and this may be at least part of the original identified TAN4 transition. Also, this protein forms dimers, and the dimer seems to be most favored around the isoionic point, which is around 5.3. So this is, I mean, again, this dimer seems, for example, it seems to be the form associated with allergy, okay? Because there are complexes with antibodies determined by X-ray. And is also relevant for milk processing. So in this presentation, I will be focusing mostly, almost only on the dimerization, and I will make some brief comments at the end about the TAN4 transition. So this being a dimerization, of course, the amount of dimer you would have in solution would depend on the concentration. You may think, okay, sure, that's obvious. But I mean, to our surprise, when we started looking at in the literature, I mean, a lot of people don't seem to be aware of the good old law of mass action. So it's really strange, because you continue to find sentences like, well, at pH X, the protein is a dimer or a monomer or something like that. When in fact, although the amount of the fraction of dimer would indeed depend on pH, and there's experimental work on that, a lot of work. I mean, the protein concentration makes a lot of difference as well, as you can see here. Let me just try to change my pointer. Okay, sure. Okay, so the way I mentioned this is that in some of the experimental results I will show you as well, in some cases there's some uncertainty about whether the form is a monomer or a dimer, because on top of the fact that sometimes it's not easy to understand in each particular experiment what was the protein concentration, but even, I mean, even the fraction, even if you could compute the fraction from the dimerization equilibrium constant, that depends a bit on the experiments. So there's some uncertainty about whether the protein is a monomer or a dimer in some experiments. And in some cases, I mean, I have to say this, in some cases it's a bit appalling, because people use completely different concentrations in different experiments in the same paper. Say, I mean, you can use 10 grams per liter in an NMR experiment and use 100 times less in a fluorescence experiment, and then people relate the different measurements as if they were referring to the same form, and they may not refer to the same form. Anyway, the method we used is is basically the same method that you saw in Miguel Machuqueiro's presentation. So it's the so-called stochastic titration method, and what it does is that it tries to combine molecular mechanics, dynamics, and Poisson-Boltzmann and Monte Carlo. As you know, I mean, you can use MMMD to sample conformations or structures, structural configuration, if you want, at fixed protonation state. And on the other hand, if you look at Poisson-Boltzmann and Monte Carlo methods, you can do the opposite in a sense. I mean, you can sample protonation states, but you are stuck in a single conformation. This is, of course, the type of methods that Marilyn showed last week. Although in her case, she has some conformational freedom on the side chains. But when I say PBMC here, I'm talking about the old way of doing it without conformers. Okay, so rigid molecules. So the idea in this method is simply to combine them, since they are kind of complementary. The idea is to combine them in a sound way. So the way it works in practice is you run an MMMD simulation. From time to time, you stop. You do a PBMC calculation, a complete simulation actually, and you get new charges, new protonation states for the molecule. As you can see here, some of these dots which represent the proton, the charges have changed here. So you get a new set of protonation states. And then since the PB model assumes an automatically relaxed solvent, right, through the dielectric constant and the ionic cloud, the implicit ionic cloud around it. After this, we do a small solvent relaxation in order to get a solvent that is relaxed as implied by the PB calculation. So in this way, you get a new set of charges with slightly different solvent. And then you continue the MMMD step, okay? And you keep repeating this over and over. After a while, you stop, you update the charges, and then you keep going on in a cycle doing this over and over. It can be shown using Markov chain theory that in principle, this is a sound way to sample. Of course, there are some assumptions behind it. This is here mostly in the case it's needed. To answer some question or something, but I will not go into detail in this. Okay. So in principle, you can show that this leads to an ergodic Markov chain that samples from a proper ensemble, proper thermodynamic ensemble, and so on and so forth. So in principle, we should be sampling conformations and protonation states all at the same time during the simulation. Okay. So. The setup for our simulation. So we use the form of BLG without any ligand, so the APO form. We simulated both the monomer and the dimer. We started with structures. If you remember when I talked about the 10-4 transition, there is this loop, this EEF loop, which is supposed to be associated with the 10-4 transition, and there are open and closed. There are structures with the lead made by that loop open and or closed. So we used both of them to get some more diversity in the simulations. So we did simulations and we did several replicates for each of them. Okay, 100 nanosecond simulations and several replicates. The pH was between we did pHs between 3 and 8. Okay. We included ions in the simulation. The way we did it is the number of ions added tries to keep the system approximately neutral. It's not exact because the charge would be changing during time because of the changes in protonation. And at the same time, to try to mimic an ionic strength of 100 millimolar. Okay. So as titrating sites, we considered all aspartics, glutamics, and histidines, and also the terminals. Okay. Moving to the results, we started by analyzing the dimer conformation, the dimer configuration, and I mean, of course, this kind of analysis is not specific of constant pH MD. Of course, this one is a strictly structural analysis. And one thing that was immediately obvious was that when you look at the dimer, you have here in this top histogram, you have the distance between the centers of mass of both partners in the dimer. And what you can see is that at the higher pH values, you have the distance is around 3.2 nanometers most of the time. Then when you go to lower pHs, you start finding peaks at lower distances. So again, at pH 4, you have kind of a mixture of everything. And then at pH 3, your peak is actually at a distance of 3 nanometers. So it's interesting because the two partners seem to get closer to each other, and that can be seen as well in the contact surface, which is found to increase as you go to lower pH. So you kind of get a dimer at low pH. You get a dimer which is more tight or more compact, if you want. At the same time, the relative positions of the two chains also change with pH. You have here a representation of one of the partners, and these cloud of points are represent the positions of the center of mass of the other partner. And this cloud here is for pH 3. Okay, it's mostly located here in this region. You have these tiny islands down here. And as you move for pH 4, you start populating the more lower region. And at pH 5, you are all here in this region, and you will remain there for pH 6, 7, and 8. Okay. So again, we see these two classes of apparently two classes of different configurations. And on top of that, the two partners seem to rotate relative to each other, the relative orientation changes. And this can be seen quite easily if you look at the interface between the two monomers, which have this nicely aligned. If you look at the X-ray structures that are available, you have these really nice aligned, these two beta strands aligned, and these two alpha helices aligned as well. And if you look at the angle between them, you see that they start to rotate at low pH values. So they become cross to each other, almost orthogonal, actually. If you look at the actual values of the angles.
Excuse me. Well, can you just clarify one thing? There are X-ray structures of the monomer and of the dimer. I mean, there's no dimer interface in the X-ray structure?
No, sorry. The structures that are available, most of them correspond to the dimer.
I see.
Okay. So sorry if I was not clear. So the example I showed here, this is a structure of the dimer. Okay. What we see is that when you go to low pH values, and there are actually no structures at low pH values, no available experimental structures at low pH. But when in our simulations, when we go to those low pH values, the two partners start to rotate relative to each other. Okay, so and you can see that if you look at if we define a dihedral angle between the beta strands and between the alpha helices, we can see that Here it's nicely aligned, okay, 180 degrees, so they are anti-parallel relative to each other, both the strands and the helices. And when you go to low pH, they start to rotate, and okay, and they reach around 100 degrees. So actually, it's a nice way to see all these effects together is to try to do a principal component analysis. Actually, you have to be quite careful in the way you do this principal component analysis, but I will not go into details. In my opinion, many of the PCAs you can see in the literature are done in a way which render them, I mean, largely useless in many cases. So one of the important things, for example, is you should do the PCA. For instance, in our case, you must do the PCA for all the pHs at the same time. Otherwise, you cannot relate them to each other. And I'm saying this because I've seen analysis like this done separately. Now, when you look at this, we again get these two populations. Okay, at low pH, you have mostly a population here in this region of the. So sorry, I didn't say. I'm showing you here just the first two principal components. Okay, the first one and the second one here in the vertical axis. If you look at the first principal component, it seems that we were lucky, maybe, and we seem to be able to capture, as you see in a moment, all the motions I've been talking about. So at low pH, you have most, this is a energy landscape, okay, so it reflects the population here along these two PCs. And at high PC1, we have mostly here most population here on the right side, and as you increase the pH, it starts to move to the left side, and it stays there at all the other pHs. So again, we see these apparently two different classes, and the mix again at pH 4. So you have this more compact configuration of the dimer, and you have the other one, which we simply call the relaxed structure. Okay, so we have apparently a compact and a relaxed state. And if you, for example, go along the first PC, you see this happening. You see as you go along the PC, the protein starts to rotate. Here they are nicely aligned. You see, you are moving along the PC1, and as you go to higher values, the proteins rotate. It's more difficult to see the approximation as well here, but it's also present. So we seem to have these two states. Actually, there is some experimental work which has been done, studies using compressibility measurements, where it seems that they have actually identified a compact state. They have assigned it to the monomer, not to the dimer. And looking at the experimental conditions is difficult to be sure about what the form is. They claim is the dimer, maybe it is, because although the protein concentration is very high, and at the ionic strengths we are working here, in our case, they will definitely be a dimer, not a monomer, but the experimental ionic strength is quite low, which can make quite a big difference in this protein. So maybe in their experiments is actually a monomer. So we don't really know if these two compact states are actually related or not. Okay. So moving now to the aspects more related with protonation and charge. So as I mentioned before, the dimerization is higher at the isoionic point. So this may raise the question: well, does charge play a role, or do they prefer to interact better at the isoionic point just because they are neutral and the interaction is easier? The usual way to look at this, I mean, in simulation work, sometimes is as you are aware of, you you just map, for example, you do a PB calculation, and then you map the electrostatic potential either on the surface of the protein, or in some cases you could draw the contours of the potential in the solution, and so on. The problem with that approach is, of course, it's an approximation, because you are using a fixed structure, you are using fixed charges, and so on. So we try to do something a bit different here. Since the protein distribution of charges actually determines the distribution of ions around the protein, we decided instead to use that distribution, to use the ionic distribution, to see what are the regions more charged in the protein. In the way, the idea here is, okay, what interests us is to try to understand for a nearby molecule what would be seen, what charge would be experienced. So our approach is simply to compute the densities of the sodiums and chlorides in the simulations, and look at them. Okay, so this is in principle more realistic, because the structure is changing and the charges are changing as well. And afterwards, we thought it would be interesting to compare it with the usual PB approach. So we used this is one of the main assumptions of the bioequal theory, of course, where you assume that the potential of unit force of the ions can be computed from the charge of the ion times the potential at this position. So you can compute the ionic distributions for the PB calculations as well. So what we found, here I'm showing this for the monomer. As expected, as you increase pH, the charge of the protein changes. So at low pH, it's mostly surrounded by chlorides. What I'm showing here are the concentration contours, okay, of 200 millimolars. So this means that the contours are enveloping regions where the concentration of this particular ion is at least twice as high as in the overall solution, okay? So at low pH is mostly surrounded by chlorides, of course, because the protein will be mostly positive. And then as you progress in pH, you end up with the protein surrounded mostly by sodiums, because the protein will be mostly negative at pH 8. At the isoionic point, around 5, you indeed have a mixture of things. So you still have regions of high concentration of ions. They are just in different positions in the protein. When you compare it with the Poisson-Boltzmann approach, I mean, they are more or less similar. The main difference is that the PB ionic densities are not as ragged as the ones from MD, and they reach closer to the protein, you see, because since you have to use a single structure, you are not taking into account the fact that on average the structure fluctuates and on average keeps the ions further away than than you can see actually in the PB case. So these densities of the PB densities come too close to the protein in a sense. For the dimer, you can see more or less the same, okay, more or less the same trends. And then the interesting thing is that we went to look at the region of the interface, not in the dimer, but in the monomer, at the region that in principle would be the interface when it finds another partner. So you have here on the left the monomer, and this grid represents the plane of the interface that it would establish with a different partner. And here on the right, you have the ionic densities of the sodiums in blue and the chlorides in yellow. And as you can see, the interesting thing is that in the upper half of the interface, you have a kind of dipolar dipolar, sorry, partition, right? So we have sodiums here on the right and chlorides on the left. So in principle, the protein is more negative on the right and more positive on the left. So what this means is that if you now imagine two monomers and you imagine that they will somehow find each other in solution and be able to approach each other. A simple way of looking at this is, okay, you can regard this as two pages in a book. If you close them, you would get a complementarity. So you will have this one interacting with this one, and this one interacting with this one. So and in the lower half of the protein, there seems to be not that much charge, although you have in the periphery, so at the interface, you have in the periphery some sodiums. Okay, so there seems to be some complementarity of the some charge complementarity mediating the dimerization, or maybe even driving the dimerization of the protein. And in fact, I mean, it is known that the dimerization is quite sensitive to ionic strength, so that's a good indication of that already. By the way, this complementarity that I'm showing here is maximum, and it's the results I'm showing here. These contours refer to pH 5, which are in fact the pH value where we find the higher complementarity here on the interface, and which is known to be also the pH where you get higher dimerization. Okay, so things seem to make quite a lot of sense. Moving now to the protonation curves of the system. So you can see here the net charge we computed for the monomer and for the dimer, and you can also see here the experimental titration curve. The experimental titration curve is shown on the dimer because considering the experimental conditions of this particular titration curve, which shows here. This is actually from sorry, I should have put the reference here. It's from the lab. It's from Timashev's lab. It's an old one from the 60s, I think. And in principle, for these for their experimental conditions, the protein should be almost all a dimer, probably. So it makes more sense to put it here on the right plot. As you can see, our prediction of the charge is quite good. There is some discrepancy here near neutrality, okay, near pH 7. But overall, the agreement seems quite reasonable. The isogenic point we determined is 5.1, both in the monomer and in the dimer, and the experimental one depends a bit on the study. It's between reported values are between 5.2 and 5.4. Okay. Again, probably for at least for the ones we could find, they seem to refer all to the dimer. Okay. Now, and this may be relevant to the discussion afterwards about the charge of the protein. We decided to look also at the bound ions. Okay. And the question here, I mean, what is a bound ion? A bound ion, you have to be defined somehow. So what we did here, we looked, the criterion we used was to assume that an ion was bound if it was within 4 angstroms of the protein. When you look at the radial distribution functions, this corresponds roughly to encompass the first 2 peaks of the RDFs. Okay, so I mean, is this a good criterion? It's not clear because we didn't see any strongly bound ions. They are there. Sometimes they go away. And what we see is that there's not a big amount of ions bound here in the simulations. They end up compensating for roughly 10 to 25% of the overall charge of the protein. So at low pH, the system, I mean, the protein and ions become less charged, right? And at high pH, it becomes a bit more also less charge, I mean, in absolute value. Okay, so there's an effect which is not that high. Interestingly, near the isotonic point, we don't see any change. Okay, and as I said, the number of ions is really small. Okay, of course, it's arguable what is a bound ion. Okay, you need some criterion either from in terms of experiments or here in terms of simulations. Now moving to the dimerization free energy. In principle, when we think about free energies, we think, okay, we will have to use some free energy method, right? Like thermodynamic integration or perturbation or something like that. But there's an alternative route, and that has been used by other people as well. And the idea here is the following. I mean, we know that the dimerization constant, or more exactly the logarithm of the dimerization constant, its derivative with respect to pH would be related to the difference of the two titration curves, of the titration curve of the dimer and the titration curve of the monomer. So you have this differential relation between the two. This is the so-called thermodynamic linkage relation. This is well known, I mean, since the work of Wyman and Turnford and Shellman and so on. If you prefer, you can look at this. If you are a physicist, if you prefer, you can look at this. This is actually just a thermodynamic Maxwell relation, okay? It's another way to look at it. This has been used for a lot of purposes, I mean, experimentally and so on. In particular, I mean, since we are talking here about protonations, and I will talk also about pKa calculations, it's interesting that Garcia Moreno used this relation in a somewhat unusual way to try to determine pKa values, which was an interesting approach. Anyway, in our case, what can we do? So we have this differential relation. So we have to integrate it to get free energies. So basically what you have to compute is this integral, okay, of the difference between the two curves. Or put in other words, you have to sum the areas, the signed areas between these two curves. So this raises a problem because in the past when this was used, for instance, with PBMC calculations by many people, I mean, in particular by Marilyn, who is here today. In this case is really nice because I mean you can do a lot of pH values. So in the end, you don't expect to have a large error because of the integration of the numerical integration. When you move to constant pH MD, things are different because I mean you have very few pH values. So if you are not careful, you may end up with significant integration errors. One of the solutions used, in particular by Janushen and Charlie Brooks, they decided to, I mean, in order to reduce this problem, they decided to use to fit to each of the titration curves either a Henderson-Hasselbalch or a Hill curve, and then to sum all those curves, and then you can actually compute the integral in an analytic way, which is quite nice. So you actually get an analytic expression for the free energy. We thought about using this, but then we thought, well, for instance, if you fit a Hill curve to a titration, curve, sometimes, at least in our hands, what we see is that, okay, it's a quite nice way of estimating the midpoint titration, but sometimes at the higher pH or low pH, sometimes the fit is not that good. So we decided to do a different thing. And what we did was, okay, we know from statistical thermodynamics that the slopes of the titration curves are related with the protonation fluctuations. This has been known, I mean, again, since if you check Wyman and Tanford in a more protein context, you can also find this. So if you have the variances, okay, the fluctuations of the protonations, of the occupancies of the sites, of all the sites. I mean, this is the fluctuations of the number of protons bound to your protein. If you know that, you can compute the slope of the titration curve. So we can do that in the simulations actually. So what you get is, I'm going back here to give an example. For instance, if you are between these 2 points, this point here and this one here for the dimer, you have one value here and another one here, but you actually can compute also the slope here from the variance and the slope here. So for each segment of the curve, you have 4 parameters. So you can compute a third order interpolation spline. And since you have a sum of these splines, you can compute the integral again in an analytical way. You just get a fourth order polynomial, which gives you the free energy. So this is a really nice general way to compute the integral using the thermodynamic linkage relation. So this is what we did, and what we get, you can see here the free energy curve that we obtained is shown here in black, okay, with the uncertainty with this gray shadow. And we show here also the estimate obtained with the hill fits, okay, with the fits to a hill curve, and also the experimental values from several different experiments, okay, at the same ionic strength that we did here, that we used, and at the same temperature, okay. One thing I must point is the following. If you think about it, I'm going back again to show you the expression. This was a differential expression, and this is an integral up to some point, which here, in this example, is pH 3. But it doesn't need to be. So what you have actually from the integration is that you have a shape of the curve. You have a curve whose vertical position you are not sure about. So what you get is a shape, both from the spline and from the hill fits, okay? So what we have done here is to say, okay, we have this shape, so we did a vertical least squares fit of the curves to the experimental points, okay? So this is what we get. We could predict that the highest dimerization is around the ionic point, again, which is in agreement with the experiments, as you can see. So the minimum of the experiments is roughly here. One thing that we realized is that when you look at a single experimental paper, the experimental uncertainties in most cases are actually very small. You can barely see the error bars in most points. But when you compare, and actually they are small enough to make possible to compute, to disentangle the values into enthalpies and energies of the reaction, because you can use the Van 't Hoff equation. But when you take the several experimental studies together, the actual uncertainty seems to be much higher. So anyway, we were quite happy with this prediction, because the optimal pH for the dimerization seems to be in the right place, and the shape, at least to the higher pH values, seems to be quite good. Although for the low pH values, there seems to be some problem, clearly. Our results, and I mean, either from the spline or from the hill curve, are quite a bit off. Okay. And you can also look at the individual contributions from different residues for this free energy, because I mean, what we did was that we integrated something depending on the titration curves, and the titration curves of the total titration curves are simply the sum of the individual titration curves. So this nicely leads to a decomposition of the free energy in terms of titrable residues. So you have components for these titrable residues. So you can compute the different components, and you can even do the spline interpolations for each of these individual curves, because we know now I mean, again, from statistical thermodynamics, you can easily derive that the slope for the individual curves would be given by the covariance of the occupancy of that site and the total number of protons bound to the protein. Okay, so again, I will not go into details on this, but again, you can compute a really nice spline, and in the end, you get a fourth order polynomial for the integral. Okay, so so we can compute the individual contributions of the sites to the free energy, as you can see here. Most of the sites do not contribute much to the pH dependency of the free energy. I mean, most of them are mostly flat, as you can see, but some of them can seem to be the main responsibles for the overall variation with pH, because the previous curve that I showed you is just the sum of all these individual curves. So, and some of the most relevant contributions seem to come from half a dozen of sites, more or less, or a bit more. And the interesting thing is that some of them are at the interface, as you would expect, right? Because I mean, this is the pH dependency of the dimerization constant. But at the same time, there are some relevant contributions that come from sites that are very far from the interface, like, for example, this glutamic 89 here. So this is a bit puzzling, and this will come again in other results. Now, here I will show you just an example of, I mean, you can look at the interface and try to understand what is going on in terms of protonations as you change pH, what protonations change, what pKa values change, how is that related to electrostatic interactions at the interface, and so on. I'm just showing you here a simple example where you have this aspartic 137 here at the interface, which is close to an arginine. You have one partner here and the other one here. And if you look at the titration curve for this site, it varies somewhat from when you move from the monomer to the dimer, and you can also follow what happens at the interface. For example, at low pH values, you even have a hydrogen bond between the two aspartates, and which is lost as they become charged. It is lost at higher pH values. I will not show you other titration curves. Unfortunately, there are no reliable, or at least for this pH range, interesting pK values that have been determined experimentally. Okay, there is an NMR study at pH values below 3.3, which is kind of useless for our interval studied here. And then there is a study which is a semi-experimental, I mean, which tries to discount those titrations using calculations and so on. So it would be nice to have sound experimental pKs to compare these with, but unfortunately there seems to be none. But so moving on, I will not, as I said, I will not go into those details, because I think there is something more interesting going on, and which is related with protonation correlations that Marilyn also already told us about last week. And to go in a bit more detailed, or in a more didactic way about this, I mean, when I'm talking about correlation here is simply the good old, Pearson's correlation coefficient, okay? And you have positive correlations between a pair of sites if they tend to have the same site, okay, both empty or both occupied. And the correlation will be negative if they tend to be different, one occupied and the other one empty, okay? Of course, the configurations of the protonation states or the microstates, if you want, I think this was the nomenclature, that the terminology that Marilyn used. Microstates that you end up populating will, of course, depend on a lot of things. I mean, it depends on whether the residues are cationic or anionic, or if they are close to each other or not, the solution pH and their pKa's. Okay. As a simple example, I show here an example with just 3 sites. So as you move at low pH, they will be all occupied. At high pH, they will be all empty. And I mean, at these intermediate pH values, you have here these microstates, okay, these 3 ones here with two protons, and these 3 microstates with just 1 proton, okay. And in principle, you would populate them as gradually as you move across pH. Now, for example, if you think about cationic, let's suppose they are all cationic, okay, like histidines and terminal and so on, and that they have similar pK values. Actually, when you have 2 protons, when the average charge is 2, actually this one is the most favored one, because here you have a repulsion between nearby sites. So what happens in practice is that this macrostate will be the favored one. So you will have, and in this microstate, you will have 2 negative correlations between the left and central, and between the central and right. And you will have a positive correlation between this one, between the left one and the right one, okay? If the sites are anionic, you get a converse situation. I mean, now what is avoided are sites sites close to each other, which are both uncharged, both unprotonated, because they will be charged, right? So the favorite microstate would be this one. When you have a combination of cationic and anionic residues and different pKa's and so on, I mean, you can get God knows what, okay? But in principle, you would expect to still have some favored or unfavored microstates, okay? Actually, when your system becomes larger, you can get these large correlation networks, okay? And in particular, you can get long distance correlations. And one thing, of course, in this example, I should point this, in these examples, I'm assuming a really strong correlation, okay? But this is just for the purpose of illustration. So when you have a large system, you will tend to be largely stuck in a sense. If you think about it, for example, think about this as a large auditorium where you have several seats, okay? And for instance, due to the current COVID pandemic, we have to have safe distances between the seats, so people will avoid sitting close to each other. So they will tend to favor either this configuration or this configuration. But the larger the system is, the larger the network of correlations is, the more difficult it is to jump from this one to this one, right? Because you have to have a lot of arrangement. So in principle, we may expect that the sites involved in many correlations would be slow proton exchangers, okay? They will slow protons slower than the other ones. And as this becomes larger, for example, when you have protein-protein association, this effect may become even more relevant. So the idea here is the following. The correlation networks, if they occur, they will end up favoring fewer protein configurations than if you didn't have any correlations, And since you are kind of stuck between a small class of microstates, in a sense, these correlations end up determining the charge distribution, you see. And since, as we have seen, for example, for BLG, the charge distribution seems to determine, or at least to be involved in association, these correlation networks may be quite determined for the association process. As I said, I'm considering here very strong correlations. In practice, the correlations would be much weaker and things will be much more diffuse. So the question is this really present in systems or in biological systems or not? Is this relevant, or is this just a really nice idea which is completely useless? So what we see in BLG is that indeed there are strong protonation correlations, and some of them are between quite distant sites. So you have here some examples. In the dimer, in this case, okay, within the same chain and between different chains, okay, with different pairs of sites, and some of them are between distant residues. We actually saw this previously, as I think I mentioned last time in the discussion after Marilyn's talk. We observed this in cytochrome C oxidase, long distance correlations, and more recently in a study with this small membrane protein of the photosystem 2, PSBS. You may argue, may say, okay, come on, why are you calling these strong correlations? They are not strong at all. I mean, at least some of them go up to magnitude of 0.4. Okay, so this is not strong, really. But as I will show you, this may actually be strong from a functional point of view. And I should also mention that, as you can see here, some of the pairs, for instance, let's look here at these two histidines. Some of the pairs can have, at some pH values, they can have positive correlations between them. At other values, they can have negative correlations, you see? So the effect of pH can change these correlations a lot. But do they form networks? Sorry, before that, you remember that I talked about the expectation we may have that these sites would be in principle slower in the way they exchange proteins, in the way that they change their protonation state. And indeed, this is precisely what we observed. BLG has 31 different sites per chain, okay? And these, let's say, half a dozen sites that are involved in strong correlations or in significant correlations, whatever you want to call them, are just a few of them. And the correlation times of the exchange of state are only, I mean, as you can see here, most sites have very, very, very low correlation times, okay? Below 1 nanosecond. And the ones that have correlation times of the protonation change higher than that are just a few ones, and interestingly, they are precisely the ones that we find involved in correlation networks, okay, or with high correlations in general. You see, we find this for the monomer and for the dimer. For the dimer, there are a few more sites, because now as you bring the two partners together, new correlations are formed, okay? There are new pairs of correlations when you bring the two partners together. But do they form correlation networks? Yes, they do. So here I'm showing you an example at pH 5. And as you can see here, you have the negative and positive correlations shown with different colors. The darker the color, the stronger the correlation is. And as you can see, for instance, one of the strongest correlations actually is the one, this blue stick you can see that comes from this corner here, up way to this one here, and this one here as well. For instance, this one here, which is very distant, is you have these positive correlated residues at completely opposite sides of the dimer. Okay, you can look at these networks in a different way using a graph. Again, you can see these were the ones that I was pointing at very opposite sides of the dimer. Is this glutamic 89 with the glutamic 89 of the other chain, okay, between the two chains. And for instance, in some cases, you can actually understand why the positive correlations arise. Like for instance, here in these three sites, okay, you have a positive correlation between these two, but this positive correlation is mediated by this one here. They have a negative correlation with this one and with this one. So this is a situation very, very similar. If you look at this in the structure, it's very, very similar to the example with just three sides that I first showed you. Okay, so in some cases you can really understand where the positive correlations come from. In other cases you cannot at all. It's very, very difficult to understand the reason for them. And one example is this positive correlation between the glutamic 89, okay, the two glutamic 89 residues. And this is actually the reason why I'm showing here correlations which are so low, in a hope to try to find the reason, I mean, in terms of the network interactions, to try to find the reason for these positive correlations. And in this case, even with a cutoff so small, it's not possible to understand why is this happening. So this is why some these correlations, although they are small in magnitude, they may be significant. You can say, okay, why significant? How do you know that this is significant in terms of the process of dimerization? And what it turns out is that it is completely significant for a very simple reason. If we look at the residues with strong correlations, or what I call strong correlations, they are precisely the ones that were most contributing to the pH dependencies of the dimerization free energy. So these correlations, although they may not seem strong, the ones that how do you say this? The ones that become markedly that are markedly different from the others, the higher ones, are precisely the ones that determine the pH dependency of the free energy. So they are functionally important. So the picture that comes out of this, at least for me, is the following. Protonation correlations are indeed clearly determined for the dimerization of BLG, because they end up since they are kind of stuck in a smaller set of microstates, they end up determining the charge distribution. And since they are the ones, or how should I say this? It's not so. No, this is not a good way to explain it. Sorry. So the way this works is that these correlation networks seem to end up controlling the pH dependence of the system. Okay, so this is why I think what I first called strong with commas. We don't need the commas. These are, from a functional point of view, these seem to be actually strong. Okay, so what is points is that sometimes, usually we look at these things and say, okay, we have an interaction between two proteins here. What are the key residues for this interaction, for the dimerization? What this seems to be pointing at is that, okay, maybe this is the wrong question. Maybe the effect is much more localized and general. And maybe we are looking at this in the wrong way. So I found this result really, really interesting. Finally, I will just very briefly show you what we observed about the Tanford transition. Okay, if you remember, the Tanford transition is a transition that is known to occur around pH 7.5. and it probably involves the motion of a loop, the EF loop of the protein. There are indeed X-ray structures with the closed lid and with the open lid made of this EF loop. So we again looked at this using PCA analysis, principal component analysis. And what we found is that the first principal component seems indeed to capture the opening of the loop. You can see here, loop started closed and now it's opening more and more. It's shown here in magenta. Okay. So as you move from low values of the first principal component, now it's closed again. It came to the beginning. Okay. And it starts to open and so on. So this first principal component seems indeed to capture an opening or closing motion of this EF loop. But can we claim that we are seeing the turn for transition? Well, maybe not, because first, the turn for transition may involve other regions. It's not still very clear what the turn for transition corresponds to at the molecular level. It probably involves the motion of this loop, but maybe something, some more things, and there are actually some interesting NMR studies on this. And on top of that, it is known experimental that at least some of the motions of this loop occur in the scale of the microsecond of milliseconds. So we don't believe that we can. I mean, we did simulations of the order of 100 milliseconds, 100 nanoseconds. So even though the actual defective time, so to speak, that constant PHMD samples, it's maybe higher than that because the protonations keep kicking the conformations, and you end up maybe sampling more than if you didn't have these kicks. But this is way too short to be ever be able to capture these time scales. So this was nice. This was a nice result, but there seems to be more to this story, especially when you look at the. You may remember that when I talked about the setup of the simulations, we started with simulations where this lid was open and with some where this lid was closed. Now, if you look at them separately, okay, if you look at the region of the turn for transition, both for the monomer and for the dimer, and the region for the turn for transition is this region 6 points, okay, pH 6, 7, 8. I mean, there seems to be they don't seem to be trapped in the original position. And if you look at the average, sorry, it's not shown here still because we were doing some analysis still yesterday evening. And if you look at it, actually the average value of the first PC, which is shown here in the x-axis, actually decreases a bit as you go from 8 to 7 to 6, both in the monomer and in the dimer. So if you think of it, as I told you, low values of the first PC correspond to closed. So as we go down in pH, the lid seems to be, on average, to be closing, which seems to be in agreement with the turn for transition. But when you look at the other pH values, at much lower values, seem to be quite strange, because clearly, especially in the dimer, the structure of the protein is still stuck in the original either open or closed conformation. So there's something weird going on at these low pH values. We don't understand why at the high pH values the structures are not trapped in the initial position of the lid, and at low pH they are trapped. They started exactly in the same way, so there's something going on here. So I mean, our conclusion seems to be right now that to study the motion of this loop and the turn for transition in general, we would need, I mean, or either much longer simulations or enhanced sampling techniques. Okay, so That's it. The conclusions. Well, about the methodology, as I tell you at the beginning, part of our goal here was to try to come with a set of analyses that could be generally useful to study protein-protein association using constant pH MD. And as I showed you, okay, we can do, I mean, the normal analysis you can do even with normal MD, like the analysis of the dimer configuration, for example. Then we can analyze the protein charge distribution using ionic densities, which I think it's a nice way to do it when you have dynamics of the conformation and of the protonation states. And I mean, it's really nice, this method that of course is not new, other people have used it. I really like this use of the linkage relation to compute the pH dependency of the free energies. This is really a nice way to get quantitative characterization of the association. Of course, you can look at the interface and try to understand things in terms of interactions and protonations. I didn't spend much time on this, largely because I think the protonation correlations seem to be quite more interesting. And I mean, at least in terms of an idea, I think it's a new thing, I think. And this can be analyzed as well. So this seems to be a nice approach to get a detailed description of protein association. In terms of the BLG, so we identified these two states of the dimer, what we call the compact and relaxed. Maybe it is related to the one previously found in experiments, these compressibility experiments that I mentioned before. It's not clear if there is response to the monomer, but maybe it doesn't. It's not related to this one. It could be a completely different thing. We also found that although the interaction, although the dimerization occurs is most favored at the ionic point, it's still mediated by charge complementarity. We managed to, as I said before, to predict that the dimerization is favored around the isonic point. And we found these correlation networks consisting of slow titrable sites. And as for the time for transition, it would have to be studied in some different way, I think. So that's it. So I have to acknowledge a big acknowledgement to my co-authors in my lab. Lucie de Roche, she did most of the heavy work. She did a lot of work, in large part because of my never-ending suggestions for new analysis in a different way and so on. She was just showing me results yesterday evening. So thank you, Lucie. And Sarah Camps, who is actually the PI of this project and whose contribution to this work is, I mean, at least as big as mine. And she had the valuable role of keeping my never-ending suggestions within a same level. And I thank the funding from the Portuguese Foundation institution. Okay, and thank you for your attention.
Okay, thank you very much, Antonio, for your presentation. I think we have some questions. Yes, there is one question from Thomas. Tom?
Hi, that was a very nice talk. Oops.
Thank you.
And very enjoyable. It turns out that since I was at an ag experiment station university, we did a considerable amount of work with beta-lactoglobulin over the year, experimental work with beta-lactoglobulin, including estimation of charge and of association constants as at pH 3. This is mimicking the stomach conditions. Okay. And I was going back and just going through it. We didn't publish a lot on this, but just going back and looking at it. And in fact, the charge that you estimate is very close to the charge that we measured. I have a poster that I can send to you that shows you some of the experimental work looking at second virial coefficient. The question we were interested in was whether we could determine the charge on a mass action association, a system undergoing mass action association.
Okay.
I presume you're working with beta-lactoglobulin A?
Yes, A. Or everything is A, yes.
Okay. Because we worked with both A and B, purified A and purified B.
Right, right. And also the experimental data I showed is all for A.
Okay, yeah.
Okay,
yeah.
so we were careful about it, yes.
Okay. The reason it makes a big difference, it might be interesting to look at the difference between A and B. The association properties are very different for A and B.
Yeah, yeah.
And it's a single valine. There's a substitution, I think it's an aspartate to a valine transition that
yeah, one of them is involved in aspartic, yeah. Yes.
Yeah. But you get the gels that form from those are completely different. So beta-lac B gives you a gel that's turbid and solid, and beta-lac A gives you a gel that is mucus-like and clear. I mean, and so for the food industry, it makes a big difference because they use it as a texturizer. One last thing. We just published a paper very recently using the fluorescence centrifuge that we developed to look at beta-lactoglobulin in milk. And it turns out that beta-lactoglobulin binds immunoglobulins in milk, and it may be part of the delivery system of in colostrum. So thank you. I enjoyed your talk very much. And I got to get your email because I'll send you a copy of the
Yes,
poster.
I would love that. Yes, thank you, Tom.
Are there any more questions? Oh, there is a question. Oh, yes, there are. So, Thomas Simonson.
Yeah, thanks, Antonio. Very interesting. Just a kind of a quick question. You showed the nice pH dependence on the dimerization free energy. And of course, as you pointed out, it's only defined up to a constant. So
Sure.
I guess if I want to use the protein to deliver, to transport something, by maybe controlling its dimerization, I would need to know if the kind of the pH variation was sort of occurring in a useful region. Like if it's varying from -9 to -4, the thing will always be a dimer no matter what. Whereas if it's varying from, let's say, +2 to -2, it's a little different. I mean, presumably these are standard state conditions, but so
Yeah.
would this be like a useful pH dependence to actually have the dimerization be sensitive to pH in more realistic conditions, not standard state conditions, and considering all the free energy, not just the pH dependent term?
Well, I'm not hearing you very well, but one of the problems with this protein is that you cannot. It's difficult to say. Well, what is the typical pH value of this system or something like that? Because, for instance, as Tom just mentioned, when the protein is in the stomach, it would be around pH 3. So even though these values here may seem to be okay, this is irrelevant from a biological viewpoint, but I mean, it isn't for some purposes. So the fact that the function of the protein is not even well understood doesn't help in this regard. I don't know if I'm answering your question, Tom, because as I said, I'm listening. I cannot listen to you so well.
Okay. Well, I guess I was really asking about the additive constant or the offset of the free energy here. How this compare? What would the total dimerization free energy look like, and would it really change much? For example, would it change signs if you went from an acidic compartment to a kind of a neutral compartment?
Mm-hmm. Yeah, I mean, the way the vertical position of this curve is sometimes computed is that you, for instance, since we started at pH 3, you can choose an experimental point and move from there, right? Start our curve here, for example, and move from there. But I don't know. So the problem is that with this approach, you cannot know the absolute value of the free energy. Maybe it would be interesting to study these using normal standard free energy methods, maybe, and try to relate them with these somehow. I don't know.
Right, but the experimental values is just normal standard state values. So that actually answers my question. Yeah.
But what do you mean by normal standard states?
Well, it's like what molar, 1 molar.
Right, the thermodynamic standard
concentration.
states. Right.
Yeah.
Oh yes, sure. Yeah, they are.
Right.
They are.
Yeah.
Yeah.
Thanks.
Okay, I see Jim has a question.
Thank you. Thanks, Walter. Thanks, Antonio. Very nice. My question, so that curve you have now, and I agree, I mean, with Tom Simmonson's question, it's, and all of us that do these, not the all calculations, but just try and work out pH dependence of energy, you have always to work out what's the bit, you know, where do we put things on that vertical axis. So I'm entirely sympathetic to the problem and spend a lot of time looking for experimental data to try and place things. My question is, so on the left here, you talked about the difference between calculated and experimental at low pH, and I'm wondering, so presumably that is directly related to the charge difference between the monomer and dimer states that you calculate relative to the actual states.
Right.
And sorry, I hope you can't hear my guinea pigs, our guinea pigs arguing at the moment. So, but that's actually not maybe a huge difference. I mean, the difference you've got there of a couple of kcals or something.
Sure.
What's that? A couple, 2 or 3? I mean, maybe it is a big difference. 2 or 3 charge units difference on either the monomer or dimer or both,
exactly.
relative to the experiment. Sorry.
Yes. Yeah. Exactly. Yeah. It will be about that. Yes.
Do you know which groups you've got wrong? Any idea what groups you got wrong? Why? I mean, I would guess you're depressing the pKa's of aspen glue too much in the dimer potentially.
Yeah, could be. It's difficult to know. You see, because for instance, if I show you if you look at the global titration curve, I mean, sure, there's a small difference here at low pH values with the experimental one, but it's actually quite small at these pH values. So the discrepancy is much larger here, right? So maybe there are some groups that are not being properly computed, but maybe it's in the monomer. Maybe it's here is fine, right? And it could be the monomer that where we have some groups wrong. Could be that, yes. And in that respect, it would be quite interesting to have experimental titration curves for the individual residues, not just pKa's, right? But individual titration curves. That would be really interesting. Because in that way, we could actually use the experimental titration curves to compute these integrals. That would be great.
Thank you.
Yeah. So I don't know, for example, if France is here today, maybe you would like to determine all the titration curves for beta-lactoglobulin. I don't know.
I think he should be here. Yeah,
I'm here. Yeah, I just have to find the buttons with the microphone and the camera. No, I was thinking about it during your talk that this would be a very interesting thing to do, also from a methodological point of view, because so far a lot of the methods that have been developed, they work really well for tiny proteins, but they have not been optimized or made suitable to study larger systems, and especially not if you get to a dimer of 2 times 18, right? And so then one needs to be a little bit smarter about it. And I was actually browsing around the website of your home institute to see what NMR possibilities you have. So I mean, maybe there is some possibility of a collaboration here to get this. And once all the bans are lifted, I'd be happy to visit and talk about it.
Okay, so we will keep in touch then. Okay,
Yes.
great.
Yes.
Thanks.
And if you don't mind, say hello to Manuel.
Okay. Yeah, I will.
Thank you.
Thank you. Thank you so much.
So Marilyn?
So that was, let's see, I don't know if you can, where I am, but you can, that was really lovely. And so one is, I was really happy that you looked at Tom, kind of did the analysis vis-a-vis Tom Lowey's thoughts, because I mean, he has really challenged us. And it's nice to see that when you get the numbers out of molecular dynamics, they're actually not so far off. And that when you compare the MD with the PB, again, they're not the same, but they're not, crazy bad. So that in itself is really a relief. So I think we can, look at Tom's stuff. I mean, maybe it'd be interesting to do it in cases where Tom would tell us the differences are larger. But this was really a nice thing. On the correlation, and I mean, one thing, if you go back to the curve where you were looking at the whole dip and the titration curve, yes, yeah. I mean, no, that one with the red dots, the experimental data, the pH dependence, yes, yeah. I mean, I'm wondering whether the errors there are due to the fact, as you were saying, that when things are more highly correlated, you have trouble converging, or you may have trouble, and then at pH 4, 3, and 4, you have all your acids titrating, whereas at pH 7 and 8, it should be many fewer residues that are undergoing titration.
Right, exactly, yeah.
I kind of have another thing, and we're also looking at correlations, as and I'm curious how you think about separating adventitious correlations from real correlations. So you're drawing a line between 2 points because they're titrating in a similar range, I think. But all the acids, if they were independent, would be titrating in a similar range, but they're not mutually repelling each other. And I wonder if you've decided that you can pull that out of the analysis of just the correlation coefficients, or if you've gone any deeper than that. I mean, we're still thinking about what to do.
I mean, the correlations we I don't know, let's.
Yeah. So.
For example, I mean, yeah, it's. I don't know. Maybe there are clever ways to analyze these networks. You have done some along these lines, right? In terms of the networks. Maybe one of the things that actually frustrated us here was it's not easy to understand the origin of some of these correlations. Ideally, I would like to, to say things like these. Okay, oh yes, there's a positive correlation because of this effect. But in many cases, it's really impossible to understand what's going on. It's a completely global effect. As we were discussing last week, Marilyn, I don't know, maybe our brain is not the right tool to think about this kind of processes. That's a bit my view. I mean, that's why we get so. That's why we tend to think, well, but this is. I think you said something like this last time. It was maybe a bit strange why evolution has, in some cases, such a, non-obvious way, right? A complex way to do things. And I don't know, maybe it's not complex, maybe it's complex to us. And this idea of trying to split away all the interactions and to understand all the things. It's not the right way to look at it. I don't know.
I mean, it's hard to know if pairwise is the right level to look at. I mean, we, you know, okay. That is certainly one question and unfortunately, once you get beyond pairwise, then the combinatorics begins to be really problematic. But right. and so for you, where you have the molecular dynamics, having two things at opposite end of the protein be really correlated can make sense if the protein changes some portion of its structure. I mean, we certainly in MCCE would miss that. Mm-hmm. But I guess I just want to check, like, if you had two aspartic acids that were really not talking to each other, they would still have their titration at the same place.
Sure, but they would not be correlated. Because I mean, if you look at some,
right, okay, I'm being right. Silly. Yeah. So they'd be. That's right. Okay.
They would be in the. I mean, in the sense that the probability of. I will be a bit technical now. The probability of having both protonated, let's say, would be just the product of one being protonated and another being protonated, because they would be uncorrelated, right? So. If you are at pH value where both of those residues are half protonated, just by chance they would have the same state 1/4 of the time, right?
That's right.
But that is eliminated when you compute the correlation. The correlation in that case would be 0, right? So. These are really, and I think this is useful maybe to stress this for people that didn't do this correlation analysis. What this correlation analysis means is that when you look at a given instant in time, let's say, they would be, for instance, if you have a positive correlation, that means that you would find them with the same state much more often than you would expect just from chance. That's what this means. So
great, thank you.
I see a question from Miguel.
Hi, so António, great.
Hi.
Once again, a very nice presentation. I think it's 2 or 3 slides before this one, the one with the energetics, before the correlations.
Right.
Yeah.
This one.
I had the same question. I think it was Thomas also. I had a question related with this. When we're looking here at his profile, it's clear that it's possible that a few residues are not really getting the correct protonation state. So when you are doing this kind of dimerization, the thing that came to my mind, because we are trying to develop this MMPBSA that also deals with pH, when I'm looking at here, it's like when you have a hammer and you start seeing nails everywhere. So my question to you is, wouldn't it be possible to do some MMPBSA where the protonation states of each conformation would be taken out of the constant pH, so you get you have the vector from the protonation state, you insert it, and then you can this way you can do MMPBSA with the pH effects, which is actually what you want to get. Wouldn't it be possible to do that?
Yeah. Well, it would definitely be possible. Maybe the way in order to combine them, maybe you should think about, maybe there's some kind of pH dependent term that you would have to include. I don't know. You would have to think about it. I mean, because now you are moving to a different ensemble. Maybe you would have to wait in a sense. I don't know, but I'm just thinking on the fly. Yeah. Maybe I'm wrong, but yes, I mean that's a nice idea. I think so.
My related question would be, because if you have such a method that is fast enough, then you could easily kind of adjust or readjust a little bit the protonation states, the vectors into, and try to find out that way which would be the residues that you would have to change that would correct the fit to the experimental
Oh, I
data.
see.
So
Yeah.
if you have something that is fast enough, you could, because you know which residues are actually sensitive and which residues are contributing more or less to the final energy. This way you could at least have a guess to see where you might be committing some sort of a mistake.
Yeah.
That was my suggestion also. Yeah.
I should also maybe point something. Let me show again. This is related with what Marilyn asked before. If we look at these plots with the correlation times for the proton exchanges, you can see. Keep in mind, our simulations were 100 nanoseconds. Okay, and as we can see here, some of the slow sites, let's call them this way, some of the slow sites have correlation times of the order of several nanoseconds. Okay, or almost going to 20 nanoseconds. So the sampling of the microstates involving these particular sites may be bad, may be quite bad, because I mean here it's not like in PBMC where you can do millions of steps, right? Here we cannot do that. So if a slide is slow, I mean then you are stuck with it. I mean there's nothing you can do to improve the sampling. So.
Oh. There is a question from Olivier.
Yes, thank you for giving me the words of. Thank you, Antonio, for the very nice talk and presentation. I was a little bit interested in this correlation analysis, and yeah, it's something in between a suggestion and a question, because sometimes, or usually, people can also calculate, in addition of just the correlation coefficient, p-value. So it's about, because in a correlation you are calculating a regression, and you can have a p-value of this implicit regression, and maybe if doing this could help a little bit in this ensemble of many interaction to like sort the correlation in between the more likely and the less likely, and maybe this could help to in the interpretation.
Yeah, that's an interesting suggestion. Yes, yeah, thanks. Yeah, that could be nice to look at it. Yeah, there's many. I mean, we are just trying to improvise as we go along in the analysis of these correlation networks, really. So any suggestion is welcome. Yeah, thank you.
Yeah, they are very easy tool to use in R or whatever to do this analysis. It's a few minutes work.
Oh yeah, sure.
Yeah. Okay. Thank you, Olivier. We have a question from France.
Yeah, Antonio. So before I already started speaking in response to somebody else before I was able to say very, very nice work.
Thank
you. Back to the discussion that you had a few minutes ago, also with Marilyn and Jim, I think. I sometimes the showing structures in my mind for these kinds of problems, it seems to be misleading, because, and then especially drawing lines between atoms in structures. Actually, I like the other graphical display that you had with the
this one. Yeah,
either with a graph or with or the other one with the little dots, because I think what matters here are other words like configuration and energy, right? I think those are the things that govern the system. It's not the structure, and sometimes looking at these structures seems to be, people get a little bit of a mechanical view of these proteins, like you push the protein on one side and then something else happens on the other side, and that's completely not how proteins work. So I think that that we should not be feel uneasy if a protein structure doesn't is not an easy object to display the data that we produce in a computation, right?
Yeah, no, I completely agree. And one interesting aspect here is, for example, it's quite interesting to compare this with the kind of systems that Marilyn was showing us last week, because I mean, in those cases, if you look at, when you look at the structure, there is implicitly, since you are thinking about proton transfer in those cases, it makes sense to think in a more, or maybe it makes more, at least more sense to think in terms of a more mechanistic way, because you know that would have to be some kind of progression of the protons along the way, right? In this case, it's completely different, because I mean, there's in principle no functional proton transfer going on as far as we know. So, but still you have these terrible intricate networks. And I agree, I mean, the way I showed this image here is just to make clear that some of the correlations occur between sites which are completely opposite positions in the proton. That's more the idea. So, I mean, the sticks here do not, of course, represent any kind of interaction or whatever you want to call it. Yeah, yeah. So they can be misleading. Absolutely, I agree.
And to the microstate problem. So, of course, many of us do bulk measurements or ensemble measurements. So, is there any? Does anybody know of single molecule ways of detecting coupled charge states? Have people done that in any way by a fluorescence technique or something? That would be kind of interesting, right?
Yeah, oh yeah. And one thing it would be also quite interesting to be able to somehow experimentally measure or the correlations themselves between different sites. But I don't see how that can be done. I mean,
well, in single molecules, I guess if there is something that optically responds to the charge state, I think it should be possible. So
maybe, yeah, that would be interesting.
Well, I will contact you afterwards, Antonio, about titration in NMR. Okay.
Yes.
Thank you.
Thank you.
There is a question from Wei.
Yeah. Hi, Antonio.
Hi, Wei.
Let's talk. It's a very quick question. Indeed, actually, you have experimental data for dimerization, pH-dependent dimerization free energy. So you have, if you seem to be happy with your dimer charge pH-dependent charges. You can backtrack your monomer pH-dependent charge. You can tell whether your calculation is good or not. Did that make sense? You have experimental data of this, and you seem to be happy with your dimer part pH-dependent charge. And you can backtrack, and then you can use this data to get the charge for pH-dependent charge for monomer, and you can check whether your calculation is good or not. Does it make sense?
I'm not sure, but how would I do that way? Because sure, I have the charge for the monomer and for the dimer, yes. And sure, I know that the pH dependence of the free energy is directly related with the difference between these two curves. Yes. And I have experimental values. Yes.
Yeah. No. Actually, sorry. Actually, I take back. I don't. Yeah. You don't think you can track? And because actually, my suspicion is that your monomer calculation may have some issue.
The monomer.
Yeah, okay. Monomer, right? Because monomer usually is much harder because it's more flexible. It's much harder to, right? Sometimes really hard to, because a lot of the conformation charge coupling is even more pronounced for monomer protein, right?
Yeah, could be. Although I mean, The structure is quite stable, actually. I mean, but yeah, could be, yes.
But the coupling, the dynamic coupling is different story. Stable or not is right. But coupling is some coupling is very subtle, right?
Yeah.
Some coupling, we cannot use our eyes to tell, but actually they occur they change the energetics quite a bit.
Yeah.
Anyway, things are pretty. Thank you for the talk. Let's talk.
Yeah, thank you.
There is a question from Tom.
Hi, it's less of a question. I've gone back. I was looking at some of my. I had an undergraduate do some work on senior thesis student. The association energy at 100 millimolar is 5.899 kilocalories, and at 20 millimolar salt pH 3, it's 4.71 kilocalories. The charge at pH 3 and 100 millimolar salt is about 13.7.
Okay. So does it agree with some of the experimental data I have here on this plot?
It's pretty close to it. If you look at this is again. I'm not sure what salt concentration you're referring to in this.
It's 0.1 molar.
10 millimolar.
No, 100 millimolar.
100 millimolar. Okay, so at 100 millimolar pH 3, it's 5.89, 5.9 kilocalories per mole at pH 3. So slide over a little bit more to the right.
Yeah.
Yeah. So it's over. Yeah. In that region, it's going to. And at lower salt, it decreases as you expect. The association decreases about 4.7.
Okay.
And the charge under those conditions, the charge at pH 300 millimolar salt is 13.7. It's plus or minus about 1.5.
Your ionic strength is which value, Tom? Sorry.
100 millimolar.
100 millimolar. Okay. Yeah. So is that those values are still not published? Is it?
Nope.
Okay.
No.
Okay.
I sent you the poster.
Great. Okay. Yeah. Thank you. Thank you so much.
Okay. So I see we are. So there are no more questions related specifically to the talk of Antonio, which I thank again. Do we want to say something? I think the discussion has already taken place from any point of view. Can we say some final words concerning how good or how bad are at present computational estimates of the charge of a protein? So we have been saying that, so we have on one side, one problem is the estimation of the titration state of tetrahydrosides, and then the way we may miss it is mostly probably because we miss the right configuration. So if we or conformation. So if we have a wrong conformation, of course, the physically sound mean that we may use to estimate the protonation state won't work. Another point that has been raised was the fact that there's apparently for proteins there are negative charges which are somehow, if not very, very strongly interacting, still they are somehow transitionally bound to the protein surface. Today we have seen that there is some correlation, some agreement between the estimated charge and the experimental data. So, do you have comments on this?
Come out fairly close. They come out within the uncertainty of both experimental measurement and the calculations. For proteins that are larger than that, they diverge, and there seems to be some, I'm not sure if you want to call it a structural element to all this, but if you take the FC fragment of an antibody. There's great crystallographic structures for FC fragment, and there's really good, very, very good charge measurements for the FC fragment. And there you're off by a considerable amount. There's a lot of anions bound. We also have data for protein where cations are bound. So it's not just anions, but it's anions more so than cations. It depends upon where you are relative to the titration curve. But there is a size dependence to it too. It seems to go up roughly as the square of the radius. So it goes up roughly with the surface area of the protein, the error in the calculation versus the measured value. And beta-lac is not too surprising. It comes close because its molecular weight's 14,000. So it's a small protein, and so it's not too surprising. It's close. It's pretty nice that it comes out to the same values that you're calculating, and the free energies of association are coming up very close to, reasonably close again to what you're calculating.
Yeah, one thing that we saw, because I mean the number of ions here that we found to be bound, at least using this criterion, is quite small actually, but I mean it's enough to bring this effect. And one thing that was also clear, which sometimes I mean in some discussions like this, people tend to neglect the co-ions, and I mean we see co-ions also bound to some regions of the protein sometimes, right? Because the protein is not homogeneously charged. So my point is this overall effect is not just due to counter ions binding to the protein, is also due to some co-ions. Okay.
I agree that. I agree with that
right,
totally.
Yeah.
Another case that you might be able to do some modeling on that would be useful would be serum albumin, human serum albumin. There's been we've done a lot of work on that. There's other measurements, estimates of the amount of chloride bound at physiological pH. It's about 5 anions, and in that case, that's a co-ion that's being bound.
Okay, that's interesting. Yeah.
Does any of the left theoretician try to use Friedrism to estimate the ion distribution around proteins to see how different may be from Poisson-Boltzmann prediction?
That's a good. I know that there were some studies. I know that there were some studies comparing Poisson. Let me think. Comparing Poisson-Boltzmann with Poisson plus Monte Carlo of ions. At least from what I recall from this Brazilian guy, Francisco Barroso da Silva. He used to work with Bo Jonsson and people in Sweden. They did that kind of comparison. Some of the papers were quite interesting, but the protein was not modeled in any detailed way in terms of structure. Sometimes it was just a sphere and things like that. But maybe they have more recent work. I don't know. But yeah, that would be interesting to know how consistent these things is.
Yeah.
Because I mean, here when we compare the ionic distributions from the dynamics and from the PB, I mean, it's interesting that the general shape is more or less similar, right? But I mean, here we used, for instance, for the PB calculations, we use a representative structure taken from the simulations and the average charges. Okay, we put the average charge on, because I mean we are computing the potential. So since the Poisson-Boltzmann equation, at least the one we are using, is linear, the average potential would be related with the average charge. So in this lower panel, the calculations are being done with the average charge on each residue. Okay.
And would you say that the ionic distribution in the molecular dynamics reached convergence? Because I think it will take time.
Oh yeah. If you ask me, okay, do you think that your 100 nanoseconds are enough? Of course not. Of course not. I mean, MD simulations are never converged. So.
Well, I guess the solvent-wise, from some point of view, is even harder, often neglected, the convergence of the ionic species in the solvent.
Yeah,
it takes some time.
And actually, the setup, and this is related again to the overall charge that you compute at the end. The actual setup of the simulations is kind of arguable, right? Because, for example, in our case, the way we did it, as I mentioned, the number of ions we used was a number of ions that would keep the protein approximately neutral, okay? Yeah. So this is a bit by trial and error. We did a few preliminary simulations to try to guess this, and at the same time, trying to keep the ionic strength at 100 millimolar. So you have two equations, neutralization and ionic strength, and you have two variables, number of cations and number of ions, and then you solve it just like that. But it is okay. I mean, this is an approximation. I think I think it was Wei that mentioned in some previous webinar that he mentioned, okay, the right thing to do was just to have a grand canonical treatment for the ions. Sure. I mean, you would choose a chemical potential for the ions, and they will go and come and go from the box as needed. That would be perfect.
But this would require, again, a long time to relax.
Probably, yeah.
Okay, any more comments? So
I have one last comment. I was able to convince or get Nils Thorstenson. I'm not sure if you know him, at Chemical Computing Group, if any of you know him by any chance. They're a company up in Montreal that develops software for the biopharmaceutical industry, looking at various properties of proteins, primarily antibodies, but other proteins as well. I was able to get him interested in looking at charge, and he went back and looked at, I don't know how many thousand crystal structures, and did find chloride sites, which you might call binding sites, on many of those. If anyone's interested, I'm sure he would be interested in having collaboration in getting the charge calculations to be more accurate. It's of great importance in the biopharmaceutical industry that you know what the charge is, especially under formulation conditions. That's what keeps proteins from clumping, from aggregating. That's a primary mechanism. And they, some companies measure the charge, but some don't. They still rely on calculations and they're getting misled. So if anyone's interested in setting up a collaboration with Nils, I can get you his email, and I know he's interested in the problem.
Okay, thank you. And Franz, I think you have also.
No, I think it's an interesting suggestion. Maybe this would also be interesting if you would be willing to present some of the data of practical challenges that people are interested to understand and compute, right? It could be inspirational for all of us. Now, I just wanted relating to the ionic atmosphere around proteins and computation of those, to just remind you of the work that Junji Iwahara presented a few weeks ago, right? In the PNAS paper that they just published last year about this, which seems a really fantastic piece of experimental data on this, and and with nice comparison to computation. I think that's a really useful piece of work in this context too.
Yeah, absolutely. Yeah.
Yeah. Okay, so let me thank everybody again. See you next week. I don't know whether the program website has been updated already. So next week there will be Walter Rocchia speaking, apparently, and so we'll meet. I think so. Soon the program will be updated until the end of May, which I believe will be the end of the webinar meetings. With some update that I will give you very soon. Okay, thanks everybody.